Just like last week, this week we're going to be looking a little bit at conserved non-exonic elements, CNEEs, which we heard last week are thought to be, at least plausibly, putative, that is, potential regulatory sequences for genes. And this week, we'll be looking at better ways to prove, or maybe more strongly suggest, that identified CNEEs really are functional elements and not just conserved regions that we don't know whether or not they actually play a role in changes in the evolution of developmental biology. And there are a couple of critical elements to understand about potential characteristics of active CNEEs, that is, regions that really would be regulatory regions of DNA sequence. We often draw DNA as a double helix, and a conserved non-coding element could be in part of this DNA sequence. Unfortunately, DNA doesn't really look like this most of the time. Most of the time, DNA, now representing the double helix as a single line, DNA is wound around proteins called histones. And this is a way that DNA is packaged into a higher order structure that is harder to break. So the DNA will wrap around these globular proteins, histones, and those proteins help package and, to a certain extent, protect DNA from physical damage or breaking, especially during cell division. And it turns out that DNA wrapped with these proteins, again called histones in blue, that entire component, before I move on, is called a nucleosome. So a histone is technically 8 different proteins, a nucleosome is technically 8 different histone proteins, so it's an octamer, and the DNA wraps around that octamer. So a single nucleosome, 8 histone proteins, and then a couple of windings of DNA around that bunch. Those nucleosomes form higher order structures, that is, nucleosome is not the primary way DNA is packaged. You can have sets of nucleosomes then that organize DNA in a cluster like that, and then there can be clusters of clusters, so that DNA gets wound into thicker and thicker sort of threads. And this is what actually forms what we think of as chromosomes when we see them at metaphase, is really this higher order winding of many, many, many layers of histones, nucleosomes, that form these really thick structures that we see as metaphase chromosomes. During the cell cycle, especially during interphase times, that is when the cell is not replicating, which is one of the times in which you see those chromosomes condense during metaphase, part of cell division, most of the time, some parts of a chromosome are double helices. They're open, that is, they're not tightly compacted into chromatin, but other regions of DNA will be more tightly packaged into chromatin. The DNA is always associated with these proteins, but it can be more loosely or more tightly associated with histones. And it turns out that the regions that tend to be condensed during interphase, during the times when genes are normally being transcribed and translated, these regions we call heterochromatin. That's DNA tightly packaged and inaccessible, I hope that's spelled correctly, to other proteins like enzymes. So for example, if RNA polymerase were here, that's the enzyme that reads DNA and produces RNA transcripts. it can't access, it can't read, it can't bind to the DNA to find genes to transcribe when the DNA is tightly compacted, bound to histone proteins in heterochromatin. But in other regions of the genome that are euchromatin, those are regions where the histones are not tightly compacted together, where the DNA is more open, then enzymes like RNA polymerase can access those regions, bind to the DNA, and start to produce, say, a transcript of a gene. So likewise, if you have regions of double helices that are euchromatin, and then regions that are tightly bound to histones, heterochromatin, if a gene is going to be expressed, not only should the gene, at whatever point in the cell cycle and whatever, and this is important, whatever type of cell, at some point in development, so this is important both in terms of time, temporal location of the cell, that could be developmental time, for example, and also physical location, that is area of the body. This gene might need to be produced, transcribed, and turned into a protein. So at this particular combination of time and place, a gene that's going to be expressed needs to be in euchromatin, uncompressed, loosely associated with these histone proteins. Not only does the coding region, the region that actually encodes the transcript and then the protein, need to be in euchromatin, but here's the critical part, any regulatory elements also have to be in euchromatin. For example, this might be the place where a transcription factor binds and recruits RNA polymerase by physical associations, positive physical associations between those proteins, recruits RNA polymerase to start transcription. So this is the key then, that those regulatory sequences that are important for gene expression should also be found in euchromatin. In other words, CNEEs, if they really are not just conserved sequences for some other reason, but if they really are regulatory, you might expect them to be in euchromatin along with, in the particular cell, time and place where a gene is being expressed, you'd expect that gene's regulatory elements also to be in euchromatin. So this correlation that you could look for between gene expression, like say feather development genes or feather important genes for this week's paper, or flight related genes, and the regulatory sequences that control them. There should be a relationship between presence in euchromatin of the gene and its regulatory sequence. There. I think I said that enough times, didn't I? There are two main ways, well, there are at least two ways, let's put it that way. For this paper, there are two main ways to understand whether or not DNA is euchromatin or heterochromatin. And this has entirely to do with how available or accessible the DNA is to other proteins or enzymes. The main way that people used to look at this was these different histone proteins have different chemical modifications to them when they're associated with loose or open chromatin structure, let's say square modifications, and tight heterochromatic structures, let's say associated with round chemical modifications on the histone proteins themselves. So now we need to talk a little bit more about histone structure. So those nucleosomes are actually an octamer, as I said, of 4 different proteins: histone H2A, H2B, H3, histone H3, and histone H4. And what you can't see on the other side of this ball on the back side are each of these again. So there's another H2A, H2B, H3, and H4, and that makes the octamer. And each of these proteins has the amino or C termini that extend out beyond the globular structure that these proteins normally fold into. So we're going to focus on a couple of these in particular. Histone H3 has at its 27th position, so let's see, that's the amino terminus, and you count 27 amino acids in. Its 27th amino acid is a lysine, which its a single letter code is K. So this position in this protein is called K27, lysine 27. And it can have two different types of modifications. If it has, again, we're talking about open chromatin structure, accessible, then this lysine 27 can have an acetyl group, AC, added to it. And that, when H3, lysine 27th, the 27th amino acid, has an acetyl group on it, that's activating, that's in euchromatin, as shown up here, that square modification, acetylation of lysine 27, associated with open chromatin conformation, euchromatin. That same lysine, though, can be modified in a different way. It could also have, instead, a methyl group added to it. And when lysine 27 of histone H3 is methylated, that is associated with chromatin that's in a closed, compacted, heterochromatic state. The same histone has a lysine at position 4 as well. So same amino acid, different spot in the protein. And it also can be methylated. And that methylation, like methylation of lysine 27, is also associated with that open chromatin structure. So in other words, if you knew which chemical modifications were on different histone proteins that were associated with different genes or different regions of the genome, you could predict, or you'd have a readout of whether or not that gene at the time you looked at it was in euchromatin or if it was in heterochromatin. So there are two ways that this can be assessed. One way is through detection of these different chemical modifications on histones by immunoprecipitation. In other words, there are enzymes, there are antibodies that are specific to these different chemical modifications. So if you wanted to know, for example, if you knew that there was a gene here on this chromosome, and you wanted to know if it was in euchromatin or heterochromatin, you could have a K27 methylation specific antibody. And so what you could do is, in this case, this is a process called ChIP-seq. This is mentioned in this paper. It stands for chromatin, which is, again, the complex of DNA and the proteins it's associated with. Chromatin, immunoprecipitation. So you're immunoprecipitating, you're grabbing using immune tools like antibodies, you're precipitating or pulling down or extracting the chromatin, the DNA that's associated with whatever that antibody binds. So if the antibody binds a histone that has a methylation mark on either lysine 4 or lysine 27, these repressive marks that cause DNA to fold into a tight structure, heterochromatin, then that antibody will pull down after, this is a critical part, first part of chromatin immunoprecipitation is making random breaks in DNA so that the double helix is physically severed into different spots, different pieces, then adding this enzyme. And so the immunoprecipitation part is that enzyme is associated with that particular sequence of DNA. So you can take that DNA and do something essentially like sequence the DNA. And so you can get a readout from a cell of all of the DNA that was physically associated with histones that had these repressive or methylation marks. And you can do the exact same process for discovering what DNA in a particular cell type at a particular point in time is associated with histones that have activating marks like acetylation on lysine 27. So the whole point, again, to review is we're trying to identify regions of the genome, which DNA sequences at any point in time in any type of cell are associated with open DNA structure. Those are probably the locations of genes that are turned on at that point in time in that cell, or regulatory elements. Other parts of the genome that aren't necessary, they're not needed to be producing genes at that transcripts at that time, or contain regulatory sequences, are possibly or more likely found in heterochromatin and associated with methylation marks on these two lysines, 4 and 27. So the outcome here can be binary. You can use either approach. Antibodies against repressive histone modifications like methylation, or activating euchromatin defining modifications like acetylation, to figure out which regions or which DNA sequences are quote unquote active euchromatin, and at the same time you can find out which regions of chromosomes, what regions of DNA are, in other words, inactive. There's another approach that can be used to identify essentially the same information, DNA that's in euchromatin or not tightly compressed, and in heterochromatin, associated with more of these histone proteins. And that is the availability of DNA to be damaged by enzymes like a transposase. And these enzymes can physically cut DNA, but only in euchromatin, or at least almost always only in euchromatin. So these transposase enzymes can't cut the double helix in regions of heterochromatin, but they can in regions of euchromatin. And if you combine these transposases with fragments of DNA that are specific sequences that are known to the scientists, if you combine both the transposase and these DNA sequences at the same time, then what the transposase does is it will break, as I drew up here. Let's add one more break for discussion purposes. So that's where the transposase has broken these fragments of DNA, and when the transposase breaks, it will add on those adapter primers to the ends of the regions it's broken. And those pieces of DNA with the ligated or attached specific DNA sequences, we'll call those adapters or primers. Then those pieces of DNA can, like in ChIP-seq, be extracted and then PCR amplified. That is, we make lots of copies of those using those primer sequences as tools and then sequence. This is another way to identify which sequences in the genome, that is, what regions of which chromosomes are accessible to the transposase at whatever point in time in whatever cell type, and that gives us a readout of which regions of the genome are in euchromatin versus heterochromatin. In other words, in regions of euchromatin, not only will we get sequences, because in regions of heterochromatin, the transposase doesn't cut, it doesn't add in these primer sequences, so we shouldn't ever see or obtain sequences from those regions, but it can also give also a more sort of qualitative view, depending on how many sequences you get from different regions of the genome, sort of a qualitative, how euchromatic or how open is the DNA, how accessible is it to this transposase. And this is the technique that's called ATAC-seq, ATAC sequencing. And this is what an ATAC-seq plot looks like. We plot chromosomal coordinate on the x-axis from position 1 to 10 million, let's say. And the y-axis is going to be the number of times a particular part of that chromosome is sequenced. And this is all based on these peaks and troughs. So there will be some regions of the chromosome that show peaks in the number of sequences or broad regions, and some troughs or valleys. And it's these peaks that are euchromatin, and the valleys then would be heterochromatin. And the euchromatin corresponds to these regions where the transposase is able to produce these small fragments of DNA that have PCR adapters, and that allows them to be PCR and sequenced multiple times, and that's why we see these peaks. Regions of accessible chromatin correlate with where sequencing is able to be conducted, this euchromatic region. In heterochromatin, you don't get any transposase activity, so those regions of the chromosome won't be sequenced often or at all. ATAC-seq, by the way, stands for Assay for Transposase Accessible Chromatin. Even though I'm writing transposable here, I'm going to correct that in just a minute. It's Assay for Transposase Accessible Chromatin, ATAC. There we go. Transposase. Good. So ATAC-seq. And we'll see plots like this in the paper that we're going to read this week.